in biological sciences from the Universidad de la República in Uruguay after teaching there for several years. He is now an adjunct professor of ethology at the same university in Uruguay, the Universidad de la República, where his research focuses on acoustic communication with respect to social and reproductive relationships in subterranean rodents, specifically within the genus Ctenomys. With more than 20 years of experience in both the field as well as the lab, he has published 16 papers and 6 book chapters related to his research. And so today he will give a talk entitled Sounds from the Tunnel, the Social Life of a Solitary Subterranean Rodent. So thank you for joining us, Gabriel, and you have the floor.
Okay, thank you. Welcome all. And thanks to the organization for inviting me to give this talk. So let's begin. Subterranean rodents are particular organisms which have an almost worldwide distribution, being adapted to underground life, even not closely related taxa, share many convergent characteristics and slightly different solutions to similar problems posed by the subterranean niche, which makes these animals popular research subjects for behavioral and evolutionary comparative studies. Underground living has some advantages like protection from predators, food availability depending on the ecosystem, control of external variables like temperature and humidity, and reduced competition. But it has some disadvantages as well that lead these animals to adapt to particular conditions like digging cost, hypercapnia, water management, or sensory and communicative challenges. Due to their underground life, they exhibit different strategies to communicate to each other using different communication channels or modalities in different combinations. One important issue to take into account is that they live in dark tunnels systems, a factor that determines that the reduction of eyes, extreme in some species, hindering the use of visual signals. But even in species that do not have reduced eyes, visual communication is almost impossible because of the darkness of the tunnels. These animals can use acoustic, chemical, olfactory and vomerolfactory, tactile, and seismic signals. Tucotucos, the Ctenomys animals, are a group of subterranean rodents endemic to the southern part of South America, Peru, Bolivia, southern Brazil, Argentina, Paraguay, Uruguay, and part of Chile. The genus includes some more than 55 species or 60 species, thought to have appeared mostly after a process of explosive speciation during the Pleistocene. While they are extremely variable at the genetic level and capable of living from sea level up to 4,000 meters in the Andes and from grasslands to xeric habitats, they apparently maintain the majority of their general characteristics of behavior, morphology, and physiology adaptations. They are adapted to subterranean life in similar ways as other subterranean rodents, but they are unique in the fact that they forage above ground during short excursions outside their tunnels. This is the reason because some colleagues consider them not as true subterraneans, but as fossorial rodents, even if in many species the time they are outside their burrows is very brief. The higher number of species found probably had their origin factors like their subterranean lifestyle, patchy distribution, limited vagility, high territoriality, low effective number, great karyotypic variability, and the characteristics of their mating systems. They are mainly solitary animals, the only exceptions being juveniles living with their mother before dispersing and pairs during courtship and copulation. But there are some social species. There is a true social species, Ctenomys sociabilis, that has been detected and proved to be social, mainly for female groups living with some males. But also there are some species considered as tolerant, like Ctenomys famosus, Ctenomys rionegrensis, Ctenomys peruanus, because they have their tunnel systems connected or they present overlapping home ranges but there is no strong evidence that they live together all the time. And there is another species, Ctenomys opinus, that has a social structure, but this social structure is labile during the day. So they are not all the day together in their burrows. Tuco-tucos use several communication channels that are efficient in an underground environment. Even if the tuco-tucos do not have reduced eyes, like other subterraneans, visual communication is impossible because the tunnels are dark. And so these animals mainly use chemical signals like body odors, glandular odors, and odors associated with feces and urine, tactile signals captured on the entire body surface and through the vibrissae, and acoustic signals using vocalizations and or mechanical sounds such as teeth grinding. Visual cues are practically absent due to the almost complete darkness inside the tunnels, but there are some indications that tuco-tucos can be sensible to UV light and eventually follow urine marks using this UV vision. Tactile and chemical signals are mainly linked to the use inside burrows. Seismic signals have not been detected in tuco-tucos like in that they do in other subterranean rodents. And acoustic signals appear to play a major role in communication systems of the tuco-tucos, and some studies have begun to explore their repertoires from physical and functional points of view. Acoustic signals, for the acoustic signals, we have complete acoustic repertoires being analyzed in two species, Ctenomys pearsoni and Ctenomys talarum. And there is incomplete information about Ctenomys flammarioni, haigi, lami, mendocinus, peruanus, rionegrensis, sociabilis, famosus, and torquatus. Then most information available on vocalizations is about solitary species. So very old papers from the 50s reported about tuco-tucos vocalizations, but attributed to them no biological significance. As I detected a lack of information about the vocalizations on Ctenomys, some very conspicuous, we decided to, in my doctoral dissertation, to study the acoustic signals of Ctenomys pearsoni in the field and in the lab, and have found 5 signal types that are described from the physical point of view and from its probable functionality. This was the first study to tackle these vocalizations using sonograms and other modern methods of analysis. And these are the detected types of vocalizations that can be divided into pulsed calls, this S-type or type S that are territorial vocalizations, type G and type J, and tonal calls that are the type C or courtship and type RN that are from POPs. The type S vocalization is a type of vocalizations that are emitted by all adults and in Ctenomys pearsoni, and the observation of animals kept in the laboratory over a long period suggests that males were vocalizing approximately twice as much as females. The common name of the animal, tuco-tuco, is the onomatopoeic representation of this sound, probably because it's easily perceived from at least 10 to 15 meters outside the burrow. So let's listen at the sound. So I hope that you heard this sound. Laboratory observations show that the animal elevates its head, opens the mouth more widely as the sound gets louder, and compresses the lateral body walls with every sound pulse delivered. In Personi, Mendocinus, Sociabilis, Famosus, and probably Torquatus, both sexes vocalize these long-range signals. In Talarum and Heigi, only males do it. In the majority of the species sampled, except for Tenomys sociabilis, main frequencies are in a band that goes from 180 to 300 Hertz and audible range. They no produce ultrasound or infrasound. These vocalizations are loud, harsh, and somewhat low-pitched sounds formed by short units, notes, called notes, delivered usually in groups called series. In these vocalizations, a note is a structure perceived by human ears as a separate sound. The series of notes are variable both in number and in timing of deliverance. Series are easily distinguishable between each other due to their silence, to the silences separating them, gaps of about 2 seconds, and a single S signal is composed by a succession of a variable number of series. The postulated function of short and long-range communications, in short and long-range communication of S signals, is suggested by the low-frequency energy concentration of the sound that possibly diminish the air-soul impedance mismatch effect, and by the marked series deliverance pattern. Subterranean propagation of sounds may impede the use of frequency modulation variation as a means to transfer information, except in close contact. Consequently, any information in S signals must be expressed through temporal patterning. Differences in interval internodes in the different parts of the signal may be sufficient to allow serious structure identification despite a moderate level of degradation due to propagation and in-between burrows. So vocalizations are interpreted as spacing localization signals based on the loudness of the sounds and on the marked aggressive spacing parameters congruent with Morton's motivation structural rules. But we have found that there are at least 2 types of long signals, S signals, or 2 formats of these signals. And we see them here. There we have a group that the vocalization consisting of 2 parts, part 1 and part 2, each with repeated notes. This is the type 1 or continuous pattern, in which part 1 is usually composed of small groups of notes, 2 or 3 notes per group, and part 2 presents a structure of single notes repeated in succession. This appears in Tenomys mendocinus, Tenomys talarum, Tenomys ruinonegrensis, and Tenomys famosus. Let's hear them. This is part 1. And this is part 2. It is somehow different. Then there is our other format of vocalization consisting of a repeated succession of groups of notes that we call it type 2 or discrete pattern that shows in Tenomys sociabilis, Torquatus, and Personi. Personi you have heard, yes, so let's hear Tenomys sociabilis pattern. It's somehow bird-like because sociabilis has upper frequencies, probably because they are social, and but they still have the same patterning of the vocalization. The possible origin of this variability could be related to random variation and fixation of those variations. It was stated that frequency, but not rhythm, is directly affected by attributes of the ecological or environmental differences between species. Then we can't attribute this rhythmic variation to environmental differences among the species. Available information suggests that type 2 pattern could have evolved from type 1 pattern to type 2 from type 1 by increasing the duration of some silences to build discrete groups of notes, and or by the loss of part 2 in type 1 vocalizations and an increase in the complexity of part 1. Another vocalization is the juvenile vocalization, is that is from individuals that are recently weaned from their mothers. Yes, and it is a sequence of the repeated pulsed guttural notes with a rhythm similar to the notes that appear in part 2 of the S-type 1 signals that we have yet seen, and with a function apparently similar to that of the adult vocalizations. Let's hear. So in Tenomys pearsoni, juvenile sounds are very similar in terms of form and apparent function to part 2 of the continuous S-type or type 1 vocalizations mentioned before. Then they could have evolved into the normal S-type vocalizations during development by partitioning of the sequence through silence modifications. In Tenomys famosus, they occur in two different development pathways. One, the series developed from juvenile precursors whose acoustic characteristics gradually approach those of adults, similar at this one, at the one seen before, and individual notes appear later in ontogeny and de novo with acoustic characteristics indistinguishable from those of adults. So grunts or G-type signals are harsh and low-pitched grunts, rather unstructured with a guttural sound, which are emitted in one or two notes followed by silence. This type of sound was emitted by both sexes when individuals confronted others in the laboratory, both in sexual and non-sexual encounters. Context suggests that they communicate aggressive disposition prior to direct attacks or in stressful situations and fight or confrontation over short distances. They sound like this. Their frequency is similar to S-type notes, maximum frequency between 1,900 and 3,500 hertz, and the main frequency is between 350 and 610 hertz. The relatively high pitch could be understood if we assume that, especially in the case of an aggressive encounter with a conspecific, a submission or a fear component may be present. It's different from S-type due to the lack of a clear rhythmic delivery pattern. It's issued by adults during aggressive encounters. They carry an agonistic message, although they can also be considered distress calls. And these signals could be a ritualization of sounds produced by accelerated respiration during agonistic encounters. As G signals seem primitive in relation to S signals due to the fact that the former, the G signals, are structurally simpler than S signals and are emitted in close encounters, S signals may be derived from G signals by the acquisition of a recognizable delivery pattern and an increment in power. Type C signals or courtship signals are present in females and males of Tenomys thalarum and pearsoni. In males are Those of males are low-intensity pulsed sound during the first phases of courtship and copulation, and those of females are sounds similar to moans, and the notes vary in number of multiple bands and in frequency modulation. Let's hear the female sounds. So their acoustic configuration is based mainly on FM sounds and corresponds to sounds that are usually associated with fear or submission. Their function sexual disposition signaling, because in Ternomis pensoni it was proven to meet different notes during courtship, and there is no successful mating or penetration without their emission. As a copulation interferes in the burrows and in the dark, where visual signals are absent, C-signals emitted by sexually aroused females seems to substitute for the lordosis display and possibly avoid aggressive behaviors of the males. Then the last type, the vocalizations of pops, then in personi and in talarum vocalizations of very young offsprings have been recorded. They are repeated notes in trains of more or less numerous notes, and they have high main frequencies and important frequency modulation. That's they are in the first days of life in demand for maternal care, are contact or distress vocalizations that appear as care-seeking behaviors, often when pups move away from the mother's nest, and mothers respond by carrying the young back to the nest and or providing them with resources. We have studied also that there is variability in the S-type sounds, and we studied this in 3 populations of Ternomis in Uruguay, Ternomis personi. These populations were Penino and Relincho for Ternomis personi, and a third from a different karyomorph, or that had differences in karyology, called Solis karyomorph. These populations were more than 100 kilometers apart from each other. 10 rhythm and 3 frequency variables were used for discriminant analysis. And the results show that a set of 9 variables allowed a completely separation of the populations. The 2 Ternomis personi populations were, Relincho y Penino, were correctly determined from one another using 4 variables, the differences lying especially in the rhythm domain, and the information about the general biology and distribution of the animal suggests that these differences could be explained as a product of chance variations and later fixations of the variants in local populations. Data suggests that the discriminant power of some rhythm variables, especially those related to construction of the series, the duration of notes and silences, and differences found in the series deliverance rate among populations, lead to the conclusion that some kind of vocal or local variants based on rhythm differences exist. So these results led us to postulate that the function of S signals as spacing and aggressive territorial signals was confirmed by the harsh quality of the sounds and the rhythmic pattern involved in the information transmission as a way to decrease signal degradation. So we think that information resides on rhythm. And we can see here that there is a coincidence in the use of temporal patterns with all seismic signals from other subterranean rodents to communicate at long distance and with a territorial marking intent. We have here Ternomis personi S-type territorial vocalization and Spalaxerenbergi seismic signal. and they are very similar because they rely on the pattern of the sounds. So, some studies suggest that the use of low-frequency rhythmic signals could be due to the restrictions imposed by the underground environment, since the propagation conditions determine that the information encoded by rhythm is more reliable than by frequency modulation. It has also been proven that a group of vocal signals emit information encoded in the temporal pattern or rhythm of note distribution and not in the structure of the notes. So tucutucos are probably part of this group, at least from the S signals that we have seen. And the tucutucos are part of their acoustic repertoire based in temporal encoded information, and probably other part based on multi-element tonal signals like the sexual signals, C-type signals. So about signal design, we have studied these signals, and we have proposed two hypotheses that could explain why the signals are like they are. One part could be due, or one situation could be due to motivation of the signals following Morton rules. So motivational restrictions cause that aggressive signals use a low frequency, harsh guttural sounds, and non-aggressive signals or appeasement or friendly relationships use quasi-tonal sounds of higher frequencies. Like in the other case, the construction of the signals could be due to propagation and the sound physics inside the tunnels, so environmental restrictions. The tunnel propagation conditions suggest that low frequencies will travel farther and be able to bypass corners, and signals are based primarily on low frequency sounds, except those that communicate over short distances. So if the code resides in rhythm, not in FM, then environmental restrictions are more limited as an explanation for signal design, and the main part of the signal design should be due to motivational parts. Some studies suggest that using low-frequency rhythmic signals could be a result of the constraint imposed by the underground environment, and that propagation conditions determine rhythm encoding information as more reliable than a frequency modulation encoded one. Other views agree with the idea that the main force determining signal design is motivation, because when we look at the signals quality, we can see that aggressive signals, like G-type signals, are mostly harsh sounds, and mating signals, like C-type signals, tend to be tonal in accordance with the frequency characteristics highlighted by Morton to define his motivation structural rules, even if they act in the same context from the point of view of propagation conditions in a tunnel. If they were based on propagation conditions only, they should be similar in their characteristics, but they are not. Then motivation seems to be the main force constraining signal design in subterranean rodents, but environmental constraint could be more influential in the long-distance signals, maybe causing these motivation structural rules to have taken a secondary role during the evolution. So for these animals that express their sociality, we can call that, in mostly during reproduction, in the reproduction period, reproduction and communication is a central problem. So in social tucutucus, like Tenomys sociabilis, the grouped sex is females, and males spend different periods of time with different group of females, fertilizing them and then changing groups. Communicating over long distances is not an imperative because they can find reproductive partners in the same group or in close neighbors. The communal tunnel systems are close to each other. While in solitary animals, each live in their burrow and they can be distant from each other depending on the density of the population in question. They need then to detect and find a suitable mate using mainly long-distance signals to obtain information about who the neighbors are and where they are located while maintaining their territories. Thus, for communication between burrows, chemical marks are limited to being deposited outside the burrow, and found by the receiver, exposing the sender and the receiver to predators. While vocalizations can be used as a long-distance signal, but require significant energy investments. In some species, only the male emits long-distance vocal signals, like in Ctenomys talar. So long-distance communication poses problems and has advantages and disadvantages because they become public information. Vocal signals acted at long distances have some drawbacks because they are potential sources of public information that could originate eavesdropping opportunities by other individuals like conspecific competitors and or predators. Long-range signals needed to reach the putative partner's burrow are also energetically costly, even if they use low-frequency sounds, and are also costlier than the chemical signals because the latter are mostly based in odors present in urine or feces. Ctenomys vocalizations can function as a source of public information and be the target of eavesdropping strategies or advertising concept strategies. But long-range vocal signals are the best way to communicate over long distances and at the same time being protected inside the burrow against predators. In Ctenomys pearsoni, vocalizations are thought to transfer information about the spatial location and of individuals, the sex of the emitter, and their reproductive condition. Females vary their vocal emission during the reproductive period with respect to the rest of the year by increasing the length and complexity of their vocal emissions at the beginning of the reproductive season and later decreasing those variables as the reproductive season goes on, probably immediately after being inseminated by a male. This could difficult their location by other males, avoiding harassment and danger of injuries by males trying to reproduce with already inseminated females, and opening the possibility to some sort of sexual selection by the female. In addition, male vocalizations that are almost invariable and emitted during all the year can be used by females for sensing the nearby males, thus being able to select the best males when they arrive to the female tunnels for courtship and mating, and also to resist non-preferred males. This is in Ctenomys pearsoni, that they have at the population level 2 daily vocalization peaks. In Ctenomys talarum, vocalizations are emitted during the twilight or nocturnal period, and in Ctenomys famosus, vocalizations were heard or recorded in the laboratory overnight due to logistic reasons and during the day in the field. In a similar way, the solitary Cape mole rat, Georicus capensis, creates sexually dimorphic patterns of drumming using their hind feet. Although similar in both sexes, foot drumming varies during the breeding season when males increase the delivery pattern compared to females. Then, again, a parallel between the seismic or drumming sounds from some subterraneans and the vocalizations here in Ctenomys. Many factors may have constrained the design of long-range signals in solitary tucotucos. Solitary Ctenomys use vocal signals capable of attaining many receptors outside the burrow. And these factors like long signals, high repetition rate, low frequencies, wider bandwidth, and power increase are the characteristics that S-type signals in pearsoni comply with. And we suppose that they are advertising individuals' condition and position for reproductive and territorial purposes. Vocalizations carry information about the spatial situation of the individuals, source location, because males move towards females apparently underground, and there are no signs of surface activities except foraging and the surveillance associated with it in Ctenomys pearsoni. Vocalizations probably carry information about the reproductive condition and sex of the sender, the signal repetition rate, and signals with more or longer series in males determine the sex. And males may be competing for territorial control and access to females. Males vocalize all year round, but make a great energy investment during the reproductive period. Females announce themselves, especially at the beginning of the reproductive season, to attract males and increase the chances of being located for mating. But once inseminated and or with offspring, they decrease in the number of emissions and the number of series per emission, probably to avoid aggression by males towards them or their young. So, solitary tucutucos would be in a communication network in which the males advertise their situation and condition towards the females and other males, and are incited to do so by the advertisements of the females. The information obtained by females in this exchange may allow them to regulate their mating among the available males, because females are induced ovulators and control the access of males to their genital tract. So, to try to tackle the reproduction and communication problem, because reproduction is a critical moment in the use of signal repertoire and is the moment in the life of ctenomys in which they become social for a time, we can say that this way, we compared two populations of two different species that have been well studied from the point of view of communication, acoustic communication, that are Ctenomys personi and Ctenomys talarum in the Mar de Cobo population. So during the initial stages of reproduction, the solitary animals should use medium and long distance signals, allowing them to locate and obtain some information about putative partners, to orientate when contacting the partner, then lowering the energy investment to reach at them, and to select the appropriate strategy to navigate towards the partner while evading predators. So these are the characteristics, or some characteristics of the two populations we studied. Ctenomys personi have a low medium density, and Ctenomys talarum high. The sex ratio is skewed to females in both. Predation is generalized in personi and on juveniles mostly in talarum. Main predators are foxes by others in personi and raptors by vision in talarum. And the activity is during the day in personi, activity outside the burrows, and day and night in Ctenomys talarum. So these animals use a reproductive signal sequence. And this scheme try to show what happens in Ctenomys personi. Energy expenditure involved in diggers opposes predation risk involved in moving above ground, and coupled with the spatial structure of the populations can determine the best strategy to contact a potential partner. Vocalizations allow to communicate over longer distances, while chemical signals, mostly aimed at olfaction, involve proximity or direct contact. Ctenomys use different signal types in sequence to gather different types of information leading to copulation, but the organization of these signals into a succession of interchangeable steps would be flexible, depending on the population's spatial structure and type of predators. So in Ctenomys personi, they begin with acoustic communication for location and sex reproductive condition being identified by the long-distance vocalization. Then, when the male travels to female's tunnel and is inside female's tunnel, chemical signals are used to evaluate again and confirm their reproductive status. Then the courtship is performed mainly using vocal and tactile signals. It's multimodal. And at last, when copulation is done, there are vaginal stimulation that induce ovulation, because tucutucos are induced ovulators, at least the solitary ones. And the armed penis of the male delivers tactile signals while copulating. So in Ctenomys personi presents a copulatory pattern of the Dewsbury 9 type, which involves no-lock pelvic movements during intermission, multiple intermission, and multiple ejaculation. And this characterizes my plus cyclic sequence goes to or makes through the possible induced ovulation. So we see here the sequence. It has a courtship period, the mounting event, and copulatory pattern that is followed by a intercopulatory pattern, and then again in cycles. But during the copulatory pattern, there are alternation of intromission with rapid pelvic thrust and deep intromissions where is thought that the animal ejaculates. And this occurs many times for each copulatory pattern. And this asks the question, that is the previous knowledge the females have of males and males from females by acoustic stimulation before, that is not very great, is producing that there is need for more vaginal stimulation to select the male. So acoustic only info about sex and resource holding power from the male. So if it's needed more vaginal stimulation, there is this cycling part of the copulatory sequence. In Ctenomys talarum, the sequence is similar, but there is started apparently by chemical signals that are deposited outside the burrows or in the door of the tunnels, just say in the tunnels, the plugs of the tunnels. Those chemical signals at that level produce or determines the location and sex reproductive condition identified by these others at short distance and vulnerable faction, thus exposing the animals to predation. But you remember that this is done by night usually, and the predators are visual, mostly because of raptors in this species. Then inside the female's tunnel, again, are new evaluations of chemical signals that are performed. Then the courtship is performed mainly using vocal and tactile signals, and there are also vaginal stimulation in this case. But Ctenomys talarum has a different model of different copulatory patterns at Dewsbury 11, no lock, pelvic thrust after intromission, and multiple ejaculations. So they go through the same phases, but you have to notice that this cycle here that occurred in Ctenomys pearsoni is not occurring in Ctenomys talarum. So the previous knowledge, the hypothesis is that the previous knowledge given by chemical stimulation outside and inside the tunnel produces that there is needed less vaginal stimulation, so less copulation cycles. So chemical probably gives info about heritable characters and kinship in this species. So much effort has been devoted to relating social characteristics with communicative complexity. And most of it has been centered on the influence of a species ecology has over the constraints and advantages of group living. Nevertheless, behavioral constraints on group living have been somehow neglected, especially when considering the role of communication could play in allowing or impairing full social grouping. Many levels of explanation have been considered to understand the causes and evolution of sociality, and various studies indicate that broader perspective is needed, as many constraints not yet accounted for could be influencing mammalian social behavior. And one element that has not been taken into account is the possibility that communication constitutes a key factor for the development of social behavior. We can think of evolution towards a sociality going through a bottleneck in which communicative complexity must be increased in order to allow higher social complexity to develop. And we tried to test this idea using subterranean rodents and acoustic signals. Sociality in subterranean rodents has been proposed to be in a continuum leading from solitary to eusocial forms. There are some hypotheses that have been proposed to explain the evolution of sociality in subterranean rodents, 4 at the time, but one of the most interesting from my point of view is the pacifistic hypothesis of Ganem and Nebo. And so we surveyed 12 species of vocalizations in social and solitary animals. Here are listed the species. And their signals were classified in 6 different categories: mating, agonistic, distress, contact, alarm, routine. The relationship between sociality and vocal repertoire were analyzed using linear regression, sociality as a binary variable, and phylogenetically independent contrast. And we combined partial phylogenies to obtain a phylogenetic tree from which study these signals. And we have these results with my colleague Christian Slage, that comparing samples of social species, 6, and against solitary species, 6, we have the following results. We have the following results. The total number of vocalizations in repertoire increased in social species with respect to solitary ones in both analyses. The number of agonistic vocalizations was also different between solitary and social species, with the latter displaying high numbers of these calls. And the percentage of agonistic vocalizations in relation to total repertoire was similar between social and solitary species, with no significant relationship between this parameter and the social structure. So these results imply that social species have more signals, thus increased repertoires by addition of new signals and or new signal categories, that the proportion of agonistic signals to total signals in the vocal repertoire is similar in both solitary and social species. Thus, agonistic vocalizations have been increased in number in social species, indicating the importance of these calls in the establishment of new relationships. As repertoire changes seem essential to cope with new and frequent kinds of interactions sociality originates, these results suggest that, at least for subterraneans, that communicative changes, especially at the level of agonistic signals, are instrumental in or are necessary condition to fulfill the path to group living. Also, a decreasing number of agonistic vocalization is expected if a species that originated as social evolved to a solitary type of life, as suggested by many authors, indicating again that the center of the communicational change still is the agonistic vocalizations repertoire. These results also suggest that agonistic signals abundance could be a good predictive measure of the frequency of interactions among individuals, thus a proxy to social complexity understood as an increased number of individuals living together. How these communication changes can occur? Well, cooperative communication system could have evolved from simpler systems like parent-pup communication signals. In the majority of the subterranean rodent species yet studied, females have an important role in raising young and communicating with them, and in regulating pup stress reactions. Tolerance difference between males and females have previously been found in Spalax erenbergi, a solitary subterranean rodent, suggesting that females are more prepared for tolerance to other individuals than males. And tolerance difference have also been found in females of 4 species of African mole rats with different degrees of sociality. These tolerance differences can be related to the ideas expressed in the pacifistic hypothesis of Ganem and Neville. And as we have said before, there is only 1 species of tuco-tuco proved as social tenomys, but there are social, like Tenomys sociabilis, but there are at least 3 species of tuco-tucos considered tolerant: the Rheonegrensis, Famosus, and Opimus, mostly for the females. All these factors suggest that females could be acting as an anchor to socialization and social evolution processes, as well as in communication evolution in subterranean rodents, by inducing social tolerance and stress reduction in pups through agonist management vocalizations, and eventually avoiding dispersal of the young. So, some final thoughts. I'm finishing. Even if the subterranean niche is a stable system, the species inhabiting it aren't necessarily as stable as the niche, and are not similar among them. In tenomys, and unlike what happens in other subterranean rodents, there are important differences both at the inter- and interspecific levels. Those differences are clear at the behavioral level, and they seem to condition the communicative and reproductive mechanisms. Those differences are determined mostly by the external surface environment, by microgeographical disposition of the animals and types of predators occurring there. And those differences could also determine local variants or adaptations in different populations of a species, like Tenomys personi. So this could be a case of phenotypic plasticity, enabling variable communication strategies in reproductive contexts that are different. So the complete understanding of the functional social structure, survival strategies in nature, and successful reproduction of telomeres is closely associated with understanding of the signal systems involved in courtship and copulation. The understanding of these signal systems is related mostly to the variable use of vocal signals in the solitary tucutuco species, and also to the context in which they were developed and in which they currently perform. So, in the end, I wish to thank all the students and colleagues that helped during fieldwork and through the development of these results and ideas showed here. I want to thank also to CSIC of the Universidad de la República and PDCIVA for funding, and thank you for listening.
Great. Thank you so much for a great talk. We do already have some questions coming through in the chat, but for anyone still watching, feel free to ask your questions in the chat. There is a character limit of 200 characters, so you might have to split it up if you have a longer question. And Gabriel, do you want to actually still share your screen just in case we have to go back for any additional questions?
Okay.
Thank you. And so just one of the first questions in the chat from Laure Chachard. So she says, sorry, I might have missed this information, but is the environment explaining why a species is social and not the other one? Do they live in the same environment?
They live, some of this, if the question is referred to the Tinomis, solitary species live in the same environment as, some solitary species live in the same environments of the social one. Mm. So probably environmental conditions do not have to do with the solitariness or social condition. Probably I haven't developed but let me go here and from this hypothesis that I have mentioned here, that explain or propose to explain the evolution of sociality in all subterranean rodents or in many subterranean rodents, these are the food aridity hypothesis that applies mostly to the eusocial heterocephalus, or apply to that. So it's not so general. The reproductive skew hypothesis that has to do with the difference that appears in social or highly social species, where there are, in heterocephalus also, there is only one female that reproduces with 2 or 3 males that can fertilize them. So in other highly social but not eusocial species, there are couples that reproduce and retain their pups to found a colony. And the historical constraint hypothesis has to do with the phylogenetic antecedents of the lines of rodents that are subterranean. But the hypothesis that is most interesting or convincing for me is the pacifistic hypothesis from Ganem and Nevo that was applied to the Spalax arenbergi, in which Nevo and Ganem sustains that there are variability in the behavior of individuals in the colonies of this animal that are solitary but can be tolerant, and that there are 3 types of individuals, genetically different or determined, that they call militants, tolerants, and intermediates. And they say that this pacifistic hypothesis says that once the number of tolerant individuals increases in a population, there is the possibility of becoming social. If the animals, the subterraneans, become social from species that are solitary, okay? So what we say is that in our work is that maybe the communication plays a role here, and that the individuals that are most characterized for playing or beginning with a socialization process are the females, because they are the individuals that raise the young and that share with the young during 2 to 3 months their tunnels. Sometimes they inherit part of their tunnels to some of their sons or daughters, and they communicate with each other. And then if some of these sons of daughters stay with the mother, they can through the donation of some part of the system, etc., they can begin a process of socialization, to say that word, to use that word, yes. So that's the answer to the question, if I remember exactly what was questioned. I don't know if this answer is the one that the speaker that poses the question wants, but is what I have to say.
So Laura did actually have a follow-up question and follow-up comment. So one of her comments was, thanks for a nice presentation. But her follow-up question was, in their evolutionary history, is the common ancestor of the Tuco-Tucos social or solitary? And if sociality is giving advantages, why would some species choose to be solitary?
Well, there is not clear, because the phylogeny of Tuco-Tucos are not very clear yet. So we don't know. There are some colleagues that sustain that or stricognats or South American rodents are social from the beginning, or were social from the beginning. But there are others that seems to think that that is not also always true. And in other groups, not only Tuco-Tucos, but other groups of subterranean rodents, like Bathyergids, that have many species that are social, it has been detected by the literature I know that the sociality seems to have surged in many different events. So they assume that original animals were solitary. But in Tuco-Tucos, we yet don't know. So what we wanted to say with these results I have shared with you at the final of the presentation, the end of the presentation, is that there is not only the environment and other factors that could be working there, but it has to be look at the communication system, because that could allow the grouping of individuals or not. And as I said, maybe I said some quickly and not understood, but sorry, the animals could be, if they came from an social origin, they could downward their repertoire of signals when they become subterranean, or they go to sociality from subterranean animals that were solitary at the beginning.
So I guess it's just, depending on the environment, it's kind of moving towards sociality or solidarity. Great, awesome. So there are a few more questions in the chat, but we are just coming to the end of our session. So anyone who is not yet able to ask any questions, or if you think of any questions later in the conference, Gabriel does have his Discord channel that you can ask questions on. And so we will be taking a quick break now. So Gabriel, thank you so much for such a great talk. I always love hearing about subterranean rodents. They're one of my favorite models.
Okay,
so thank you so much. So for our viewers, we're taking just a quick 10-minute break. So tune back in at 3:10 GMT for some more talks. Thank you.